Right, it's a real pleasure to be here and an honor to be opening Grape Days 2024 from a science perspective. And I mean, my first slide was really about why grapevine improvement, but I don't think we need to dwell on the negatives too much, given that, as Philip's just said, there's a lot of challenges facing this industry, and hopefully I can give you a little bit of the excitement around how we can use modern research and modern science to tackle some of these challenges. And in grapevine improvement, you're really talking about a long-term output. Nothing's going to change in terms of the genetics we have next year, but the opportunities we have are far more dynamic and short-term than they've ever been in the past, which is good because we've got more pressures, or at least a higher rate of change than ever before. You can see in this slide on the image on the top left, the mean annual temperature against the long-term average is progressively getting warmer in New Zealand, much higher than it ever has before. We're seeing more frequent and severe weather events like spring frosts, and a need to reduce inputs like irrigation and mildew applications. And all of these provided not only a need but an incentive to develop faster ways of adapting a plant that wasn't actually ever selected for its performance in New Zealand, wasn't bred for its performance in New Zealand. We've got a few genetic types of this plant that perform very well, but we plant them largely as a monoculture. We plant a clonally reproduced crop, and as we've seen in the case of PSA and kiwi fruit, and more recently in the Fusarium wilt and the Cavendish bananas, that also leaves you quite exposed to any change in the environment, be it environmental, climatic, or a new pathogen. And generally, in the case of PSA, the solution to that was new genetics, finding a resistant variety and being able to quickly replace that susceptible material with resistant material. So at BRI, grapevine improvement encompasses a few different types of projects. That includes accelerated breeding, the import of disease-resistant material, which I won't dwell on too much today because Jim's going to talk more to you about that directly after this talk. Understanding how we can use new techniques to accelerate greater gains, genetic testing to understand the differences between the clones we grow and optimizing those, and just distinguishing between varieties. We maintain the National Vine Collection. We work on rootstock improvement. Our largest piece of work is on improved Sauvignon Blanc clones, and then we have another project called Tune Vines, which I'll tell you about towards the end. So if we're looking at grapevine improvement, or any crop improvement indeed, you've got four options. One is traditional breeding, crossing plants with one another, and that means taking generally a variety which is performing very well, an elite variety, and crossing it with something that has a trait of interest, and often that's a wild relative if you're trying to introduce resistance. The problem with that is that you produce a plant that is 50% of each, and so to recover all the elite traits from that primary variety, you have to go through several rounds of back crossing, and in something like grapevine, that usually takes 3 to 4 years per generation. So to get to 5 to 7 cycles of back crossing, you're usually looking at 2 to 3 decades of work, and that's directional. So if you do all 3 years in that to achieve an outcome, and you want something slightly different, you basically got to repeat the process from the start. The other option is mutation breeding, and this is basically taking advantage of the raw material that drives evolution. Evolution is a combination of random genetic variation and then a selective pressure that picks some organisms to survive and propagate more amongst the future populations. And so we can actually increase the rate of genetic variation. People have used radiation and chemicals in the past, but the problem is then you've got a random set of diversity. So you have to then take a large population and select the individuals from within that population which have benefits. It does have the advantage of not needing multiple cycles, so it's much quicker. Transgenic breeding, trans meaning across, so that's taking genes across usually the species barrier, and that's been very successful. The majority of the world's maize, canola, cotton is currently transgenic plants. Obviously, those plants are then GMO, and so that has barriers for uptake in the market and certainly legislative barriers and regulatory hurdles in New Zealand. And then finally, gene editing, which is a much more modern technique that you might have heard about under the acronym CRISPR. and that means changing the genes that are already there in the plant. And this is sometimes GMO and sometimes not, depending on where it's done, depending on whether you actually leave any foreign genetic material in the organism that's been edited. So in each case, the raw material, let's see if we might be lagging a little bit. Could you advance one? There we go. The raw material for genetic change is diversity. Diversity is just organisms that are different from one another. And so there's two types of genetic diversity that are relevant in viticulture. One is diversity between varieties, or intervarietal diversity, and that's what you know as the Sauvignon Blanc versus Merlot. This is a much greater scope of diversity because these plants originate from a single seed. So every Sauvignon Blanc plant tracks back to a single seed that was selected, and the same is true for Jazz apples or the Sun Gold kiwi fruit. That includes new varieties, new hybrids, so that all the DRVs have to be sold under a different name, and that's important because the name that goes on the bottle is a strong indicator of quality in the market. Obviously, there's a much greater scope of diversity then. Within varieties, there's also diversity, and this arises as a result of random genetic changes that accumulate over time. All organisms get them, we get them. It's the reason people have birthmarks or different eye colors, but it happens at a much slower rate, and the scope of genetic change is smaller because usually you're seeing one gene change at a time. And that's what we know as clones in the industry. So they're much more similar. It could be the production of these clones could be natural or artificial, but the diversity is far more limited, and that's why we don't usually describe it on the bottle. Right, I'm going to show you a quick slide about our program. Over the last year, the Bragato Research Institute has been establishing a new grapevine improvement team. We're based here at Lincoln University, where we're tenants, and we have a new laboratory that does a lot of genetics, a lot of molecular work, understanding the differences between different varieties of grapevine and different clones of our premium variety, Sauvignon Blanc. The goal is to produce new individuals of Sauvignon Blanc that show traits that will put us in good position for the future, traits such as improved disease resistance, higher yield, and the ability to adapt to a changing environment. Plant and Food Research is our major partner in this research program. They've been doing a lot of the plant production for us, and also they have expertise in plant breeding and a group of good research scientists to contribute in that way.
DNA sequencing will help us to find out if there is any variation between the clones we are testing, and we compare those sequences against the parental clone as well, and that will allow us to find out if there is any variations and what that meant for the Sauvignon Blanc program.
So we're using two different approaches to identify the plants which show really good potential. One of those is driven by the plants themselves, and so we're screening for lots of different phenotypes under lots of different conditions, and plants that look interesting we can then go on to characterise in more detail what's going on at a genomic level. So one of the starting points for us is to generate a really good reference genome for Sauvignon Blanc. The genome is about 600 megabases in size, and what we're trying to target and what's achievable with the technology that we have is a genome in which all the chromosomes are assembled in one piece from end to end.
Bragato is a third generation, the most advanced sequencing equipment that's available currently in the market that will allow us to sequence long DNA fragments. That makes the difference to get the outcome that we wanted to see in a timely fashion.
So over the next 7 years, we plan to produce a large collection of around 12,000 plants, and the goal is to have a collection that represents the full scope of diversity that's possible in Sauvignon Blanc. So plants that produce higher yields and more disease resistance or interesting flavors and aromas. And then from within that collection, we want to do a few primary selections of plants that actually are showing the expression of those new traits. Currently, all of the material we grow is genetically identical because it's propagated by cuttings, and it's performing really well. But we know that we have a changing climate, and that means that we're going to need material that can handle future challenges like spring frosts or less water. So that's one of the key goals of this program. All right, so where do we get clonal diversity from? Well, traditionally, the main source of clonal diversity is, as I said, just natural genetic diversity that occurs when plants when plants grow, they have to divide their cells, they have to copy their DNA, and there can be errors in that. They're getting it constantly irradiated from the sun, and so these genetic changes can damage the DNA or cause small changes that occasionally affect traits. This is the type of thing you'd look for in the vineyard, and typically large clonal collections have been collected by people going out into very old vineyards in Europe and occasionally in North America and sampling vines that are often over 100 years old. We don't have vineyards that old, so instead we're working with our industry to try and get as much, to take advantage of the broad spectrum of the same genotype that we have growing around the country. And so these are a few images that have been collected from members of our industry. If you ever see something that looks really unusual, that's what we want to know about. So we've got a program called Oddvine. You can just go to oddvine.co.nz on your phone, snap a photo of any vine that looks unusual. It will ask you for the GPS coordinates. So if you say yes to that, put in a few contact details and why you thought that vine was interesting. From that, we can then develop a repository of genetic variation. I've got a few of these tags here that you can just apply to the plant as well so that we can find them easier. There's a QR code which will take you straight to the Oddvine website. So I really encourage you, not only those of you here, but also the people in your company, viticulturists, people working in the vineyard, just try and spread the awareness, because anything that looks interesting, be it timing of bud burst, color, ripening, senescence, anything like that is really valuable to us. There's another source of genetic variation that we can take advantage of, and that comes from understanding how plants increase their diversity. Typically, it's very slow, and so that's why you go looking in very old vineyards. But tissue culture, which is basically propagating a very small cutting of plants, when we propagate plants for vines for planting, we're taking a large cutting. Tissue culture is essentially taking a very small cutting, just a few cells in some cases, and growing up whole new plants from that. And tissue culture itself puts a stress on the plant that can lead to a higher rate of genetic change. And so plants have this very nifty ability to adapt to a changing environment by increasing their genetic diversity. Typically, tissue culture is used to produce a lot of plants from a very small amount of starting material, or in the case of grapevines, to produce virus-free material from virus-infected plants, which is what you're seeing in these two images on the left from University of California, Davis, and on the right at Geisenheim University. And to prevent them from going into the state where they produce a larger rate of genetic diversity, they maintain the conditions very, very carefully. They maintain the temperature and the humidity of these plants. And so we can take advantage of that by actually doing the opposite, by giving these plants that are in tissue culture a brief period of adverse conditions, of stress conditions. And in response, they begin to adapt, because plants can't run away like animals do, so they remove their silencing that prevents them from accumulating genetic change. And so what we do is we take these plants in cell cultures, we stress them, we then recover plants from those, and the new plants have these random genetic changes. And so what we're producing is effectively new clones of the same starting material. This is what it looks like. In the top left, you're seeing a picture of plants growing in tissue culture. They're growing in a tub that has a nutrient medium. The next step, they have to be deflasked. So they go out into potting mix in the nursery when they're very sensitive, but from there on, they've been growing really robustly, and now we have, together with our research partners, Plant and Food Research, we've produced over 6,000 new Sauvignon Blanc clones that are growing in the nursery, and the oldest of these are now sort of head height for me. They've grown really well. They've been transferred to these 18-pint bags. Each one has a unique QR code, and we're tracking them. In the bottom right there, you can see the type of interesting phenotypes we're seeing. Some have different leaf margins and leaf shapes, different growth and vigor, and this coming winter we'll standardize them by two budding them. The next big challenge for us is selection. So this is random diversity. It's not directed. These plants aren't GMO because we haven't introduced a gene that we want, but we're mimicking and accelerating the evolutionary process. So we now have to apply the selective pressure that evolution usually would. Fortunately, there's some ways we can add efficiency to that. So we use genetic sequencing as a first pass to understand what's changed in these plants genetically. That involves removing a leaf from each plant, purifying the DNA from them, and then sequencing them in the lab on the sequencer here. And some of you might have seen some news earlier in the year that we produced a reference genome for Sauvignon Blanc, and that involved a whole lot of deep sequencing and assembly to understand the genetics of mass select, or UCD1, the genotype that we're beginning with here in New Zealand, so that we have something to compare all these new plants with. We've also sequenced the commercial clones of Sauvignon Blanc so we can understand how they differ genetically and how this drives trait differences, and also so we have markers for telling them apart. Another type of selection is to actually take a trait-based selection. One of the primary traits of interest that our industry has identified is mildew resistance. That's obviously a hard one to screen in a collection of plants where you only have one of each. You don't want to go inoculate the whole collection with powdery mildew. Fortunately, we've been working with Cornell University and US Department of Agriculture, who have developed an automated approach for screening plants for powdery mildew and downy mildew resistance. And what they do is they've built a robot that has a set of very advanced optics on it. And this past season, we sent them leaves from 200 of our new vines and some of those from the National Vine Collection. They then take a standardized leaf disc from each of those leaves, they inoculate them with the mildews, and then after allowing them to grow up, they put them into this robot which scans all of the leaf discs and uses a machine learning algorithm to digitize a score for inoculum growth on all of them. So using artificial intelligence and analyzing a very large number of images, you can screen plants for their susceptibility to mildew, and we've got some very promising variation with regards to mildew showing up in the plants already. We're also looking, as I said, to import varietal diversity to get in some of the resistance genetics that's not in New Zealand. I won't talk too much about this because, as I say, Jim's going to, but these are some images from a trip to Germany where we were with collaborators looking at a variety of different genotypes that we could import to New Zealand in the next coming few years. And then finally, we've got this project called Tune Vines. And this is looking at taking some of the genetic knowledge that we have to actually implement it within seasons to vines that are already in the ground. One of the challenges with grapevine improvement is that to change a plant's genetics, you have to replace that plant. So the genes are set on the whole within the plants once it's planted in the ground. We know that we have seasonal variation. We know that we have a rapidly changing climate. And so this grew up from a desire to be able to use the knowledge of which genes are causing expression of different traits as a management tool from year to year in the vineyard. And so while all of the plants that are Sauvignon Blanc mass select in New Zealand have the same genes, which genes are turned on at different times of the years or in different parts of the country leads to the different trait expression and partly are a significant contributor to what we know as terroir. So one of the members of our team over the last year and a half went and collected DNA from plants across New Zealand and looked at something called epigenetics, which are these molecular switches controlling which genes are switched on and switched off. And what she found was that if you compare the vines across New Zealand, that they separate quite distinctly based on which of their genes is being switched on and switched off. And she's now looking at short-term pressures such as disease and drought as well. And the reason for this is for us to find genes which we might actually want to intervene with. So if we could switch on or switch off genes intentionally, then perhaps we could delay ripening or prepare plants for a stress event such as a drought if we're going to turn off the irrigation for a period. And so in parallel with this, another member of our team has been looking at how could we modulate these genetic signals. And the image on the left there shows the dream would be to have something that we could spray onto the vines within the season to trigger the switching on and switching off of genes. Fortunately, there is a molecule within grapevines that turns on and turns off genes, known as double-stranded RNA. And so she's been working on how to get RNA targeting specific genes into grapevines if it's just applied externally by spray. And on the right there, you can see a couple of images of fluorescently labeled molecules that we've managed to get into the grapevine. And so this is a really exciting project together with Scion, PFR, and Lincoln University. It's just been progressed as a very large Endeavour bid to the second round of the Endeavour funding. So that's something we hope will be going ahead later in the year. So finally, I'd just like to acknowledge the large amount of funders supporting this work, New Zealand wine growers and the Ministry of Primary Industries, who fund our Sauvignon Blanc 2.0 program, including a large set of grantors, as shown here, including Constellation and Indivin as our platinum grantors. I'd like to recognize our research team based in Lincoln, who 2 years ago we didn't have a team there, and they're just such a fantastic group of scientists. So if any of you want to come down and visit, we're very happy to meet with you and talk more about the work. Also, you can go to our website, on the BRI website, have a look at the Grapevine Improvement Program, get our contact details, and learn more about what we're doing. And there we go. I'll hand over to Jim.